So, hello guys, my name is Mohammed Naem. I'm a second year medical student, and today I'm going to give this lecture about the excitation and contraction coupling. Okay. Uh, this is my name, this is my email, this is my WhatsApp number. Please feel free to go for it, to contact me if you have any questions about this session. Okay. So yeah, by the way, just call me Naeem. It's better, okay? I'm way too used to be called Naeem and for you guys to call me Muhammad, so just call me Naeem. Okay, so let's start. For Okay, it's lagging. Okay, now it's back. So just for the introduction, guys, this is the normal cardiac muscle cell, as you guys all know. You took it in MSK probably. It looks very similar. This is the transverse tubule. Okay, let me just put the pointer. I think it would be better. Yeah, this is the transverse tubule. This is the sarcoplasmic reticulum, which is instead of the which is basically the muscle version of the endoplasmic reticulum. This is the sacrolemma, which is the basically the cell membrane of the cardiac muscle or the muscle cell. These are the M line, the H zone. We're gonna go into all that. I think this is very clear stuff. I'm just gonna move next, okay? It's just like a pure introduction. So let's start talking about the ultrastructure of the sarcomere. As you know, guys, this is a sarcomere here, as you know. For the sarcomere, guys, we'll have two, like for each myofilament that makes a sarcomere, it's made of two main integrating filaments, okay? We have the A band, the thick filament, or the myosin. You need to know all the three names. All the three names might become in the come in the question, and you read what is the A band. No, you need to know what is the A band. It's not always going to come as myosin in the question, and it's not going to always come as the thick filament. The doctor might just literally tell you the A band, and you have to know it. So the A band, which contains the thick filament, which is mainly made of myosin, as you see this one, the red one, is basically the myosin, which is the thick band. On the other hand, the thin band, which is the second myofilaments or the second filament, the second filament that makes the myofilament, which is this one, the actin, which is the I band, actin, thin filament. They all have the same name. Okay, I think you guys went over that a lot in MSK, so I think that is very clear. I'm not gonna waste more time on it. I just want to make sure you guys know this because your doctor really emphasized on this, which is that the thin filaments are attached to the Z disc. You know, this is the Z disc. It's called the Z disc because it literally looks like a Z. Okay, super clear. I mean, it's very clear. On the other hand, the myosin is organized by the M line. Okay, guys, I need you to know this is the M line, okay? I mean, this is the myosin. The myosin need to be held in its place. You remember back in MSK, the good old days when you were taught that the myosin does not move. It's the actin is the one that is moving during the contraction. So the M line is basically in the middle of the myosin just to keep it in place. It holds it in place so it does not move. Just imagine if we don't have this M line keeping the myosin in its place. Each contraction or each cross bridge that's going to happen, the myosin is going to keep moving and moving, and we won't have a normal contraction. Is this clear, guys? I think this is very clear. One other thing that I need you to know, which is hexagonal arrangement. Do you know what this means? Do you guys know what does this mean? Or do I need to draw it for you? What does a hexagonal array or a hexagonal arrangement mean? So, okay, 0, 6. Yeah, but yeah, it is 6, but how 6? It is 6. Okay, let's just draw it. Let's just draw it. So let's get a pen. So guys, basically we have the myosin in the middle, okay? You see here how we have the myosin in the middle, and we have 6 actin filaments around it. Just remember like a flower, okay? So this is the myosin, and we have 1 actin, 2 actin, 3 actin, 4 actin, 5 actin, 6 actins around it. So each myosin is surrounded by 6 actin filaments. This is what they mean literally by the hexagonal arrangement. Is this clear? It's just a very simple concept, but Dr. Simon, for some reason, emphasized on it. So I think you really need to know it. But yeah, should I move next? Clear? Okay, clear, clear, clear. Then let's move next. Okay, so this is the thin filament. Let's start talking about each one of them individually. The two filaments that we literally just talked about. So the thin filament, or on the other name, the actin, or the I band, is made of three main components. You already know that from MSK, but I really need to go back into it, which is the G actin, or the actin monomer, the tropomyosin, and the troponin complex. Again, you guys took that in MSK, so I don't want to really go into details because it's already late and you guys are probably very tired. So yeah, basically for G-actin, it's the normal actin molecules, it's called actin. So this is literally the monomer of actin, okay, which binds to the myosin for the whole cross-bridge interaction. You remember that from MSK? Tropomyosin is this strand. You see this strand, this this orange strand here. This is the tropomyosin. It's basically a long protein. It's like a filament. And for each tropomyosin, we have seven actin units bind to it. Do you need to know this number? Yes, you do. Why? Because it could come in an MSQ. Why is it relevant? I actually don't know. Do you need to know it? Yes, this is how medicine is. So this tropomyosin is literally this filament, and we have these 6 actin molecules. There are 7 amine actin molecules. They bind to it. Okay, this is still till now very clear. You got to remember that obviously the thin filament, or the thin filament structure is double strand. So as you see, this is a 1 strand. This is 2 strands. So we have 2 tropomyosin with actins and with the troponin complex. So 2 strands. You got to remember this number because in the thick filament, it's different. It's not 2 strands. Okay, so let's talk about now the last one. So we talked about the G-actin, which is the normal monomer. We talked about tropomyosin, which is the long filament. And now we need to talk about the troponin complex. So why is it called a complex? It's called a complex because it's made from more than one subunit, right? Yeah, it's just basic knowledge. It's called a complex because it's made from more than one subunit. As you see here, we have 1, 2, 3. Let's go to the next slide. So yeah, so let's get the pointer. So you see, guys, we have 1, 2, 3 subunits. As you guys probably already know, and you probably obviously studied this in MSK, but we gotta go again into it. We have troponin C, troponin I, and troponin T. All 3 of these, they all are they eat like in the complex. We have 1 subunit of each one of them. So we have troponin C, troponin I, troponin T. They are all in the same, they are together forming this complex. Okay, so you need to know obviously their role. Just to make it very simple, troponin C, it's C. So it's gonna bind to what? To calcium. That starts with the C. That's why we call it troponin C, because it binds to calcium. Simple. Troponin I, on the other hand, remember when we said that, like in the last slide, I just said the actin basically binds to myosin in the cross-bridge reaction. I said that in the last slide. If you remember, you should remember. Yeah, it's been like 1 slide. So basically, what we have, if actin is always exposed to the myosin, our muscle is going to keep contracting, and we're going to die. Our heart is not going to pump blood efficiently. All muscles are not going to work efficiently. So yeah, it doesn't work like that. We cannot just keep always contracting. So what we have here is troponin I, which binds to the actin. I don't know here why the doctor did not actually write where specifically it binds to the actin. That's something that you really need to know. Basically, this is the actin molecule. It has a specific binding site to the myosin, right? So not the whole actin binds to the myosin. It has a specific binding site. This troponin I comes and bind to that binding site. So now the binding site is not free for the myosin to bind in it. So when myosin comes, it does not bind. So no cross-bridge happens. So that's why we call it troponin I, because it inhibits the binding to actin. I inhibits, same letter. Again, that's why we call it troponin I, because it inhibits. We have also troponin T, which binds to the tropomyosin. We have one that binds to the actin. So we're going to have one also that binds to the tropomyosin, because remember, troponin is just like, after all, this whole complex, yeah, it has a lot of roles, but it's still a structural protein. It still has to do its part in the structure, like making the structure of the filament. So yeah, is this clear, guys? Are these three laws, these three roles clear? I think it's pretty simple. You guys probably took it in MSK, but yeah, you gotta know it. Clear? Okay, just these numbers, the molecular numbers, I don't think you need to know them, صراحة. I mean, if I were you, I would not study them. In my opinion, just know that troponin T is the largest one, just to be safe. I don't think Dr. Simon is gonna ask you about this. Okay, so everything about the thin filament is clear. Should I move next? I guess silence to mean everything that clear. Okay, let's move next. Okay, okay, yeah. So we talked about actin, so we gotta talk now about what? About myosin, obviously. So this is myosin. This is not the whole thick filament. This is just the myosin molecule. Remember? Let's go back. This is the whole this is the whole thin filament. This is not the whole thick filament. This is just the myosin. So for the myosin, basically the thick filament mostly contains of myosin. Each myosin molecules have a tail and two heads. So this is one myosin molecule. It has a tail and it has two heads. You see how it has like basically a tail and two heads. Okay. Okay. So the heads, they are identical subunit, and they are basically, it's where we basically the place where it binds to the actin. Remember, we said we have an actin, on the actin we have a myosin binding site. It basically binds to the head of the myosin, and here it says it's the site of ATP hydrolysis. Guys, remember back in MSK when we were talking about contraction, whatever, and we said that the muscle contracts, but when it needs to relax after the cross bridge, we need ATP in the myosin. We need ATP hydrolysis so we can have energy for the relaxation to happen. You remember that in MSK? You should, do you remember that we use ATP? Yeah, you guys remember? Okay, sure. So basically, where do we use this ATP? We need an ATP. Where do we hydrolyze the ATP? You probably know from mole that you need to hydrolyze the ATP so you can get the power, the energy, right? So the hydrolyzation of the ATP comes here is here in the head. The doctor puts a line under it, so it is important. So that means know it. Simple, I think. Nothing too crazy about this. The last thing in this slide that is just telling you that the thick filament is made about 300 individual myosin molecules packed together. You see how 300 myosin molecules. Remember, in the thick filament, we said we only have two strands, not two actin, we have two strands. Here, it's saying that we have 300 myosin molecules. So you see, I think you need to know these numbers. I don't know why, but they ask about it. I remember that we got a question on, I think we got a question about, literally, I think we got this in our midterm. So I think, yeah, you need to know about it. Again, we did not, as you said, thick is not only myosin. So thick is not only myosin. I said that thick is not only myosin. It consists mainly of myosin. When did I say that thick is not only myosin? And it's not, صراحة, it's not only myosin. There's other components that plays a role into it, but you don't need to know it. I don't even know them myself. They're not mentioned in the lecture. Just don't bother yourself. There's one component that we're gonna go on literally in the next slide, which is titin. Literally, we're getting into it. Tamam? Is there any other questions, guys? You're welcome. Any other questions? Everything clear about this slide? Okay. So remember, guys, when we said 300 myosin molecules, see, this whole thing is one thick filament. See how many heads we have? We have so many. All of this is just one thick filament. So let's just read it together. Each myosin is, this means that they are together, tethered to the thick filament by its head. And yeah, again, we said, guys, remember we said that the head is for the binding. So obviously the head is going to be sticking out. You see, this is, okay, let me just use the pen for a sec. Okay, see, guys, this is the thin filament, right? And this is the thick filament. Why is the heads out like that? So it can bind to the thin filament. If it was in, there would be no binding. So the cross bridging won't happen. Clear? I always do this صراحة because when I used to study the cross bridging, I always remember it like this. I'm not sure if you guys want to remember it like this, but it's really helpful for me at least. Myosin molecule are arranged that cross bridge stick. Yeah, we said this. Each filament at the center. Yeah, we said this. I explained what is the hexagonal array, right? You guys by now know what it means. Basically, one myosin and it has six actins, six actin filaments or so, yeah, around it. This is like the normal arrangement. By the way, the doctor said this a couple of times and he mentioned it like 4 times in the lecture. So that means it probably coming in a TBL and an exam and a midterm and a final. So know it. It might actually come as an SEQ. He brings like a picture of the sarcomere and tells you what is the arrangement of the thick filaments with the thin filaments. You have to say hexagonal array or hexagonal arrangement. Lose my dad money, to be honest. So yeah, let's just move. Okay. Now, whoever just asked me that the thick is not only myosin, well, here we comes. We have another. Okay, let's just put the pointer back. I actually really like the pointer. So we have titin, which is this molecule that, as you see here, it goes from the M line to the Z disc. Guys, just one advice: when you study, and there's a picture, please don't just read the lines and never look at the picture. The picture just makes it 10 times easier. So look, this is the titin molecule. It goes from the Z disc to the M line. You see the picture, now you memorize it. Reading it, you'll be like, you might read it and memorize it for the day. Next day, you don't remember it. But the picture is going to always be there. So you're in the exam, you have a question. The titin goes from what to what? You're going to remember the picture. You're not going to remember the typing, right? So yeah, basically this titin molecule, as you see here, it goes from the M line. Remember what the M line is? The one that holds the myosin. And the Z disc, the one that is attached to the actin filaments. You see these actin filaments? They are attached to the Z disc. Titin basically get attached to both. So so this is the titin, I get attached to both. As you see here, guys, you see how it's like coiling here? Why do we need that? Does anyone have an idea? Sarahatan is just like literally written in front of you, so I'm not gonna wait for the answer. I'm just gonna give you the answer. So guys, this is the muscle, right? The muscle contracts, but sometimes it stretches too, the muscle, like right now, my biceps is stretching. It's not contracting, right? It's fully stretching. So why the muscle does not tear when it stretch? Well, the titin plays a role in that. Obviously, not the whole job, but it definitely plays a role. As you see here, it's like cold here. Remember, like it's like a spring. Remember, guys, when you pull a string, how like it adds more length with less tension. So as you see here, it basically give you length to the muscle, as it acts as a spring. This confers passive tension as longer. Basically, yani, when you push it, like when you stretch the muscle, it helps you with the stretching that the muscle does not tear. And when you stop the stretching, it calls back to the normal length size. This is the whole idea, and less obviously like less tension because it's not a straight line. If it was a straight line, the tension is going to be like, let me just get a paper. Okay, guys. If the muscle was like this and there was no coiling inside the filament, the tension is going to be very strong when you pull it, right? When you relax or when you lengthen the muscle. But if it was coiled like this, and you start stretching it, see how you don't feel the tension directly because you still have like place to stretch it. That is the whole idea of the titin. Okay, is this clear, guys? Please, you need to know this concept because we're gonna like get back into it at the end of the lecture. Is this clear? Okay. Yes, it is clear. Okay, nice. It feels good for getting a response. Okay, so yeah, this is our first checkpoint. This is the question. We have 2 sets of the integrating myofilaments they are composed of. Very simple. صراحة, you should know how to answer this even before CVP. Well, yeah, does anyone have an idea about what is the answer? See, that is true. The thick filament is the myosin, the thin filament is the actin. Specifically, guys, I wrote this because you need to know the names. You need to know that the thick one is the myosin, and you need to know that the thin one is the actin. You need to know that the thin one is the I band, and you need to know that the thick one is the A band, because they don't like, it's not like they're gonna bring you one name. in the same question, they might say the thick filament and actin. I don't mean like they are the same name, but like they bring like in the same question, they could use two terminology. So you gotta be clear with that. So obviously, okay, that thing is lagging. Yeah, yeah, this whole hearts thing is our amazing vice head Tala creation. So yeah, I did not put those hearts, believe me. But yeah, let's move next. So now we get into the actual part that I need you guys to focus about, which is the reaction. Okay, reaction. I mean the interaction, the actin-myosin interaction, and basically the cross bridge and the contraction. So guys, let's get the pen. I like the pen here. So we have the troponin complex, as we said. We have the tropomyosin. We have the actin. Is this clear? It should be clear. For the tropomyosin, we said we have C, T, and I. Now we're gonna use them. Okay. So guys, one main concept that you need to know: any contraction when it comes to muscle, the main ion that we're talking about always is calcium. Calcium is like the biggest thing when it comes to muscle contraction. It is the number one ion that you need to know. Why? Because as we said, we have in the troponin complex, we have troponin C. What is the role of troponin C? It binds to calcium. Why does it bind to calcium? Does it just bind to calcium for that? For like no reason? Obviously, no. Well, when it binds to calcium, the troponin complex, it changes. The structure of the troponin complex morphologically changes. How does it change? Remember when we said we have troponin I and it's covering the myosin binding site on the actin? So this is an act where's the cursor? Goddamn. Wait, where's the cursor? Okay, here's the cursor. So yeah. So this is the okay, this is getting tough. Okay. So this is the actin molecule, right? We remember we had a myosin binding site. So troponin I is binding to that binding site on the normal time when we don't want the muscle to contract. We have the troponin I binding to that binding site. So the myosin does not bind to the actin and the contraction happens. Well, when calcium binds to troponin C, the whole complex morphologically and structurally is going to change. How will it change? Basically, the troponin I is going to move from that binding site. So now we have the actin, the myosin binding site on the actin, troponin I. Troponin I is basically going to move when calcium binds to troponin C. Is this clear, guys? Is this clear? How it moves after binding to calcium. We probably took that in MSK too. Yeah, it's clear. Nice. So when the binding site is exposed, actin binds to myosin. You see this is myosin and this is actin. They bind together and the whole reaction happens. Right? I think that is very clear. Just gonna move next. Okay. Okay, now we need to get back into the architecture, and now I need you guys to focus with me. We're gonna do some drawing, and I forget everything that is written in this slide. I need you to focus with me 100% on this. Okay, so we have the T tubules and the SR. Very two important structures you need to know. Like this is like ABCs of the cardiac contraction, like ABCs of physiology in the cardiac, like in cardiophysiology. We have, as we said, the sacromella, which is the C, which is the cell membrane, right? From it, we have transverse tubules coming down. Okay, so this is the transverse tubule. Let's focus now on the transverse tubule, or what we like to call the T tubule. Again, two names, the same structure. So the first thing that you need to know about the transverse tubule or the T tubule, it's its location, where it goes. You need to know this for some reason, but you need to know it that it runs above the Z disc. Remember we said when we talked about the Z disc that binds to actin. Remember the actin bind to? Well, the T tubule literally runs over it. You see, this is the Z disc here. And let's get the pointer for a sec. See, this is the Z disc, and this is the T tubule running over it. Okay, is this clear? This should be clear. Now, what is the job of this T tubule? It's not a structure that we have there for no reason. Obviously, it has a job, it has a function to do. Guys, as you know, muscle cells cannot create their own action potential. Action potential comes from the nerves, right? in like most cases. Obviously, like we have the SA node and all that. That's like different case. But no, we're talking about like the ventricle muscles or the ventricle cardiac muscles. They need they need the action potential to come from somewhere else, right? So, or not even the cardiac muscle, even normal muscles. They need the action potential to come from the nerves, from the fibers, from whatever. So basically, they come from the action potential comes from outside the cell. How do we get it throughout the whole cell? As you see, the cardiac cell or the muscle cell is huge. So we need to get the action potential to reach all its parts. So when the cell is contracting or like the filament is contracting, it all contracts at the same time. Just imagine the heart, each part of the heart contracting at different time. Just imagine how the heart is going to pump. It's going to become like this. And the pumping obviously is not going to be official and we're not going to be alive. That's something we don't want, obviously. So yeah, we need these transverse tubule to transverse this action potential throughout the whole muscle cell. Okay, is this clear, guys? Is this concept clear? That's why we have the T tubule to transverse action potential. Is this clear? Okay, I would take that as a yes. Let's move on. Well, let's get into the bend back. So this is why we have the transverse tubule. This is the sarcoplasmic reticulum, right? Which is basically the ER version of the muscle cell, the sarcoplasmic reticulum. What is the main function of the sarcoplasmic reticulum and muscles in general? Does anyone know? Okay, I will tell you. Basically, have all the calcium inside it. So the whole calcium that we need for the contraction, exactly, that is true. It stores calcium. So basically, the whole calcium that is in the muscle, it is stored in the sarcoplasmic reticulum. We need the calcium to be released for the contraction and the cross bridging to happen. Remember what we said on the last slide? So sarcoplasmic reticulum stores the calcium. You see here, guys, please focus with me. You see in this part specifically, the T tubule, how is it very, very close to the terminal cysteria of the sarcoplasmic reticulum? Like it's almost touching it. It's not physically touching it, but it's almost touching it. Why is that? So we said the transverse tubule have the action potential running into it, right? This action potential is going to give a signal that we're going to talk about later to this sarcoplasmic reticulum. This signal is going to tell the sarcoplasmic reticulum, please release the calcium that you have stored in. Once the calcium is released into the cytoplasm, the whole cross-bridge reaction is going to happen. So is this clear, guys? You really need to know this. What is the T tubule? Where is it going? And what is the function of it? What is the sarcoplasmic reticulum? Where is its job? And why the terminal cysteria of it is very it's almost attached to the transverse tubule. Again, it's not physically in touch, but it's very, very close. You see how close it is? The signals. You have an action potential, right? Remember, any contraction needs an action potential to come. So if we're talking about the biceps, the nerves that goes to the biceps, the motor nerves that goes to the biceps, will the motor nerves that goes to the biceps will have the action potential, right? Will have the action potential, it goes to the biceps. It basically transverses the action potential. The action potential goes through the transverse tubule, through to the muscle cell. Basically, you see, to the muscle cell. So calcium could be released from the sarcoplasmic reticulum. In the case of the heart, remember you guys took the SA node, just Tala just explained it to you. The SA node and the Purkinje fibers and all of that. Did you guys take that or not yet? You guys take? I think you guys took that. Basically, yes, you took that. And you took that the SA node produces its own action potential, right? And it sets the base of the heart. So basically what happens, the action potentials from the SA node, then to the AV node and the Purkinje fibers, bundle of His, all of that. This action potentials that goes from these fibers, okay, reaches the outside of the cardiac cell, right? So the action potential then runs through the transverse tubule. So through the transverse tubule, as you see here, the action potential comes from outside, as we said, from these fibers or from the neurons in the case of skeletal muscles, but in the case of the hearts, basically from the SA node, mainly from the SA nodes and all the fibers that conduct the action potential from the SA node. It gets into the transverse tubule. It runs all the way in the cell, like around the cell, as you see here. We have 1, 2, 3. They're all going all around the cell. and they get the action potential to the sarcoplasmic reticulum, give the signal there. So the sarcoplasmic reticulum releases the calcium. Is this clear now? Do you get from where the signals come? 100%? If you guys did not understood it, I would just repeat it again and again and again because this is really crucial. Okay. And I think everything's clear. So let's just read everything that is written in the slide just to make sure we did not miss anything. So as we said, the T-tubule ones on the Z line, again, I don't know why you need to know that. صراحة, I don't know. And when I studied it, I was tilted. Why do I need to know that? You need to know that, period. Just know it. You just know it. It's medicine. The T-tubule are continuous with the surface. As we said, it comes with basically the surface membrane of the cell, which is the sarcolemma, contains extracellular fluid. Yeah, just remember, since it transfers the action potential, it's going to have an extracellular fluid. So the action potential can run through it, right? Just like basic logic. True? I think that is, yeah, just know that it has an extracellular fluid coming into it, is made up of the sarcoplasm is made up of longitudinal and terminal elements. That means like this is the longitudinal elements, okay, and these are the terminal ones. The terminal ones are the ones that are like very close to the transverse tubule, okay, and these are the longitudinal elements basically. Just like the two components. صراحة I don't think you're gonna get asked about it, but just know it. It's on the slides. The terminal cysteria of the SR is located very close to the T-tubule. Again, I emphasized on that here. I emphasize on it again. They are very close. So when the action potential runs inside the T-tubule, the signal directly goes to the SR, binds into it, so the calcium could be released. That's why the terminal cysteria is basically this. You see this yellow stuff, which is the end part of the sarcoplasmic reticulum that is very close. This is the terminal cysteria. If you don't know the meaning of this term. The arrangement is crucial for the excitation. Yeah, as we said, so basically all this arrangement that we have been explaining for like the past 5-6 minutes is just for the calcium to get released so the muscle can contract. Remember, calcium binds to the troponin C, troponin I, actin binds to myosin. Remember that? I just explained it like literally one slide ago. So yeah, should I move on? Is this whole concept of this arrangement clear, guys? Okay. So we move on. Everything is clear, so let's move on. Okay, now this is literally the same thing that we explained, but we have 2 new terms that you need to know. Okay, let me just get this out of the way because let me get my face out of the way. Okay, yeah. So this is, guys, the T-tubule. You see this? See this structure? This is the same as this. It's just from a different view. And this is the SR. You see this is the same as this. Okay, let's just get the pointer so it's more clear. This SR is the same as this yellow structure. And this T-tubule is the same as this blue structure. Okay? So basically we have, as we said, the T-tubule, which has the action potential running into it, right? And the SR, which stores calcium, as you see here. So you need to know 2 things, basically 2 things from this slide, and they are very crucial to know. You're gonna get 100% asked about them. 100%. No way that the midterm or the final wouldn't contain 1 question at least about these 2 channels. The first one, which, okay, screw the order that, okay, I shouldn't say that, but let's not move with the order that the doctor put. Let's just move with here. Let's start with the DHPR one, okay? So the DHPR one is located on the T-tubule. So this is the T-tubule. This is the DHPR receptor or the DHP receptor. It's located on the T-tubule, okay? It's an L-type calcium channel. Tala just like less than an hour ago explained every single type of channel. So I expect you guys to be good at with that. So basically what happens here is there is action potential coming, okay? The action potential acts on the DHPR receptors, okay? On the DHP receptors. When it acts on it, this L-type calcium channels are gonna open, okay? And when they open, the calcium is gonna flow to inside the cytoplasm and to the basically to the and close to the SR. Is this clear? So again, action potential comes inside the T-tubule. The T-tubule has the DHP receptors. when the action potential acts on these the DHP receptors, the DHP receptors are going to open their L-type calcium channels, and the calcium is going to get inside the cell. Okay? Okay. So this is for the T-tubule part. Now let's talk about the SR part. We said that there is a signal that comes from the T-tubule and goes bind to the SR, right? So what is the signal? It's basically the calcium that gets in. Remember, we just said that the calcium channels open and calcium gets in. This calcium that gets in is the calcium literally. Remember, okay. This is remember, this is the T-tubule, okay? The T-tubule have extracellular fluid. So technically, we're still outside the cell. Not outside, but we're still not into the cytoplasm, okay? We're still not in the cytoplasm. So when this calcium channel opens, the calcium is released into the cytoplasm. And remember, the cytoplasm contains the SR, the sarcoplasmic reticulum. So the sarcoplasmic reticulum, so this is the whole cell, okay? We have the cytoplasm. Inside the cytoplasm, we have the SR. And the T-tubule is literally just passing by. And when the DHP channels are stimulated by the action potential, the L-type channel opens and the calcium get inside the cytoplasm. Literally, it goes inside the cell, inside the cytoplasm. Is it clear now? 100%? Okay. So this calcium get inside the cytoplasm. Let's go back to the pen. So we said this is the THP receptor. It opens. Now calcium is getting inside the cell. Okay. Some of that calcium is gonna act as the signal that affects the SR, the sarcoplasmic reticulum. What is the job of it? It contains the calcium. Okay. So the calcium that we just got from outside the cell, from the T-tubule, goes and binds to the SR. Or where does it? No, wait. Where does it bind? It binds at the ryanodine receptor. So it binds here. You see the ryanodine receptor? This one. I think the name you guys the name is familiar from MSK, but you definitely need to know this one. It's crucial. 100% you need to know this one. So the calcium goes and bind on this receptor. When it binds on this receptor, the sarcoplasm, the SR is gonna open, and the calcium that is stored inside the SR is gonna get pumped out to the cytoplasm of the cell, and this is how you get the whole calcium concentration inside the cytoplasm for the whole contraction to happen. Remember, we need calcium for the contraction. So is this clear, guys? This is a crucial concept that you need to know. Should I do it again? Should I go over it again, or it's all clear? I yeah, because the SR is calcium also from the SR also. Wait, so calcium from the SR also, like you have the calcium inside the SR, right? Where is the SR located? The SR is literally located inside the cytoplasm. So when it's open, the calcium is going to get outside to the cytoplasm itself. So both the calcium that is from the T-tubules and the calcium that is from the SR, both are going to go into the cytoplasm. Is this clear? So again, action potential runs inside the T-tubule. The DHP receptors are going to open. Calcium is going to get in. Some of that calcium is going to bind to the ryanodine receptor. After it binds to the ryanodine receptor, the calcium stored inside the SR is going to open. The calcium inside the SR is going to get pumped out to the cytoplasm of the cell and go binds to the myofilaments for the whole contraction to happen. Should I move next? I think I should, since you said it's all clear. Okay, now we have a checkpoint. We almost reached half part of the lecture. Almost half part of it. So is everything clear till now, guys? Till now, we're just talking about the basics. So if anything is not clear, please tell me so I can go and repeat it. Everything is clear? Okay. So everything's nice, right? You don't need any help. I mean, صراحة we all need help, but I hope at least in this session everything is nice. So since you guys all said everything is clear, so I'm gonna move on. So this is just couple. I mean, just couple of. Okay, this is just like couple informations that you need to know. Salaha is like just pure memorization. We just talked about this. The relationship between the DHPR and the ryanodine receptor. Basically, what the doctor here wants you to know that in skeletal muscles, you see here, we have more DHPR. Okay, we have more DHPR, and like it's completely organized. You see how here how it's organized? Like each one has each DHPR has one ryanodine receptor, and they are all completely organized together. This is how is it in like the skeletal muscle. When it comes to the cardiac muscle, we have less DHPR. Okay, so in the cardiac muscle, less DHPR, so we have fewer calcium channels. Basically, this is and like it's like literally random. Like there is no specific places that you are assured that this DHPR is there. So this is the T tubule. See, it's randomly. Suddenly it's here, suddenly it's here, suddenly it's here. It's like random. On the other hand, when it comes to the skeletal muscle, it's very organized. One here, one here, one here, one here. Is this clear? It's like just pure memorization. Sarah, you just need to know whatever the doctor wrote on the slide. Nothing to completely explain. One thing that the doctor really emphasized on in his lecture is the physical connection in the skeletal muscle. The ryanodine receptor 1 with the DHPR are physically connected. So they are like this. They're literally physically connected. When it comes to the cardiac muscle, as we said, they're very, very close. You see here, they're very, very close, but they're not physically connected. So they're not touching each other. They're just like very close. So the calcium get inside the cell, and some of that calcium bind to this SR or to this like ryanodine receptor for the whole SR to release its components. So just know skeletal muscle, there is physical connection. Cardiac muscle, there is no physical connection, just pure memorization. صراحة, I don't think there's any concept here to explain, so I'm just gonna move to the next slide. Okay. Okay, let's move on. Okay, now the tough part of the lecture. Now this is the part that I know, guys, it's late now. I know that most of you have been like listening for this for us talking for like the past 2 hours, or like now it's like 3 hours, but this is the part that you guys need to really like focus on. صراحة, if you don't get this concept, you did not study this lecture. So please, please, please focus on. Okay. So again, this is the T tubule that we just talked about. This is the T tubule. Okay. That has the action potential coming through it. Okay. Okay. Let me just get my pen. I think pen is here better. So this is the T tubule. Literally, action potential is coming here. The action potential is coming from here all the way to here. These are the L-type calcium channel that we talked about. So this is the DHPR. So the DHPR is here. Okay. And these are the calcium channels that are related to the DHPR, right? So the action potential comes all the way here. They stimulate the DHPR receptor, and the calcium channels open. And now calcium is getting pumped inside the cell. See, this is the cytoplasm. You see, this is the whole cytoplasm. This is the whole cytoplasm. And this is the SR, okay? So yeah, this is the whole cytoplasm, and this is the SR. Remember when I just got, someone asked me does the calcium get inside the cytoplasm? Yes, it does. As you see here, some of the calcium goes to the cytoplasm itself. Some of the calcium binds to the ryanodine receptor on the sarcolemma. So we're gonna focus about that. See here, we don't have physical connection. Calcium is getting in and it's binding to it. This is in the cardiac muscle, in the ventricle muscle. Look, calcium is getting in and it's binding. There is no physical connection between the two receptors. So when calcium binds here, the sarcolemma, basically the ryanodine receptors, calcium channels open, and the calcium is released from the SR. Okay, so we have two sources of calcium to get the concentration that we want. One source is the calcium channels, and one source is the sarcolemma. Not the sarcolemma, the SR, the sarcoplasmic reticulum. Is this clear? Is this clear that we have two sources? It is clear. But one thing also you need to know that is very, very crucial. So from the T-tubule, from these type of channels, the L-type channels that are related to the DHP receptor, from it we get 30% of the calcium. So you see all the calcium now that we have in the cytoplasm for the contraction to happen, 30% of that calcium is from these channels, from the DHPR. So basically from the T-tubule, from outside the cell. Okay, 70% of the calcium is from the sarcoplasmic reticulum. That's why we have all this pathway, because most of the calcium is inside the SR. We need to get it outside. Okay, so just know the numbers: 30% from the T-tubule, 70% from here. Clear? These numbers, guys, are very essential. 100% you're getting asked about it. 100%. I saw the recording of your lecture. The doctor like said them a couple of times. When the doctor really repeats something, he's asking about it. So he's gonna ask about that. Okay, okay. So the calcium right now is in the cell. What does the calcium do in the cell? Basically goes to the myofilaments and the whole reaction happens. The whole cross bridge happens. Actin get exposed, they bind to myosin, and the whole reaction happens, right? So now we have our, now the contraction happened, right? But we still have the calcium inside the cytoplasm. We cannot keep the calcium inside the cytoplasm, obviously, right? We need to get it out. We need, some of it needs to go back to the SR, and some of it needs to go back to the outside, right? Just logic, because if calcium is inside the cell, two things are going to happen. It's going to keep binding to the actin, and the contraction, the heart is going to keep contracting, which will lead to death because you don't have ATP in the heart as much, and the heart is going to become ischemic and die. That's a long story. Just to make it more simple, remember, calcium does so many things other than binding to the myofilaments. One of the things that it does, calcium actually activates a lot of the enzymes inside the cells. So if the calcium concentration is high for a long time inside the cell, a lot of enzymes are gonna get activated inside the cell, and they're gonna start like basically destroying everything in the cell because the enzymes are hyperactive. That's why you need to get calcium outside of the cell. That's why actually in normal cells, calcium concentration inside the cell is very low, because if calcium concentration is high, the enzymes are gonna get activated, and they're gonna destroy the cell. Basically, the cell is gonna kill itself. So we don't want that to happen. We want the cell to live. So yeah, so now we have 100% of the calcium from inside the cytoplasm. We said 30% from that is from the outside and 70% from the SR, right? So logically, we need 30% of this calcium to get outside again and 70% to go back into the SR, right? Pure logic. So let's talk about, first of all, let's talk about the 30% because it's simpler. You remember this channel that Tala just told you about, basically the sodium calcium exchanger, the one that she said I'm gonna explain? Well, it's the time I explain it. Basically, what you need to know: 3 sodium get inside of the cell for 1 calcium to get outside of the cell. Okay, this is the whole concept. Basically, it's an exchanger from its name, sodium calcium exchanger. 1 calcium get outside the cell. This is calcium. See, this calcium get outside the cell, and 3 sodium get inside the cell. Okay, so we said 30%. Does this contribute for the whole 30%? No, it contributes basically for 28%. So 28% from the whole calcium we have in the cytoplasm is pumped outside the cell by this sodium calcium exchanger. Is this clear? Do you need to know 28? Yes, you need to know this number too. There's a lot of numbers you need to know in this lecture, which is kind of annoying. Well, yeah. Is this clear, guys? The 28 number thing. Okay, I would take silence as a yes. So 28 is from this. So there's still 2% we need to get outside the cell, right? Which, like 2% to reach the 30%, and the rest we're going to get back into the SR. That's a different story. But now we're talking about the 30%. So 28% we get out by the sodium calcium exchanger. We have like 2% left, which this ATPase. So this thing is called calcium ATPase. Basically, from its name, it's an ATP, so it's going to use ATP to get calcium outside the cell. 2% only. Why only 2%? Because it's slow acting. like it's very slow when it comes like the sodium calcium exchanger is like very rapid because it does not need ATP hydrolysis and whatever, so it's like very rapid, just like concentrations moving, just ions moving in and out. For the ATPs, you need to get the ATP hydrolyzed, and like that takes some time. Basically, that's how صراحة, I don't know if that's the reason, to be honest, but this is how I just memorized it. It's slow because it needs to break down the ATP. So it's calcium ATPase. It takes out like it takes 2% of the calcium outside the cell. So now the whole 30% that we got from outside the cell from the T tubule, now we got all again out, right? 28% by the exchanger, 2% by the ATPase. Is this clear, guys? Are the two channels clear? Do you want me to repeat anything about that? Okay, they're clear. Tamam. So now now 30% is outside the cell. We still have how much? We still have the 70% of calcium inside the cell, which is the 70% that we got from the SR. Obviously, this calcium we need to get back into the SR for the next contraction. Remember, it's not just one contraction and the cell is done. Remember, the heart keeps pumping, so the heart needs to keep contracting. So this calcium needs to get back into the SR. Guys, remember in MSK when you took circa 2? You guys remember the molecule called circa 2? You, I hope you remember it because we're gonna talk about it right now. If you don't remember it, don't worry, I'm gonna explain it literally at this moment. Basically, circuit 2 is this. You see where he's you don't remember circuit 2. Okay. Basically, circuit 2 is a molecule. It's an ATPase, literally an ATPase that is like it's this one. You see, forget about the the PLP. This ATP, it's circuit 2. It's a molecule that is called circuit 2. What is the job of this circuit 2? It basically takes calcium from the cytoplasm and pumps it back again into the SR, right? So the 70%, how do we get it inside the SR? By a molecule called circuit 2. It basically uses ATP to get this calcium and pumps it back into the SR. Okay, you get the job of it? The circuit 2? Is the job of the circuit 2 clear? Yes, okay, it's clear. طيب, so since it's clear, remember guys, nothing in the body is just like doing one job. There's always inhibitory. There's always something that inhibits it. Because if like basically the metabolism or like the function is going only one way, the cell is gonna die. If all the calcium that we have in the cytoplasm always is pumped back into the SR, there will be no calcium ever in the cytoplasm because the circuit is working rapidly to get this calcium back into the SR. So we don't need that. I mean, we don't want that. So what stops it? We have, do you see this PLP? It's basically a molecule that's called phospholambin. I hope the phospholambin is probably a new molecule. I don't think you took it in MSK. Basically, what does this phospholambin do? Okay, now I need you really guys to focus with me. Please look at me and focus with me. This is the circa. Imagine this is the circa, okay? The circa molecule has 5 phospholambin binding into it, right? Each molecule, each phospholambin, when it binds to the circa, what happens is when it binds to it, it literally makes it work slower, right? So now if we have a circa with no phospholambin with no phospholambin binding to it, the circa is gonna pump the calcium rapidly, maximum rate. Now we have one phospholambin attached to it. So the pumping is gonna be slower. 2 attached to it. So it's gonna be even slower. 3 attached to it. It's gonna be even slower. 4 attached to it, even slower. The 5th one attached to it, the circuit is almost not working. So there's almost no pumping back into the circuit. I mean, into the SR. Is the phospholambin concept clear? I think it's so one circuit surrounded by 5 phospholambin. Please repeat. I will repeat. Okay, guys, remember we have circuit 2. This molecule, what is the job of it? It takes this calcium from the 70% that we released from the cytoplasm and pumps it back into the SR. This is what circuit does. Remember, every pathway in the body, or like every action in the body, will always have an inhibitory method to it to stop it. Because if the circuit keeps pumping calcium back into the SR, you would never ever have a contraction, because the calcium could never have like good because calcium would never reach the concentration that we want it inside the cytoplasm. So we need something to stop the circuit when we don't want it to work, which is the phospholambin, right? The phospholambin is the molecule that inhibits the circuit. How does this phospholambin works? We have one circuit, okay? Each circuit is surrounded by 5 phospholambins, right? Is this clear? One circuit, 5 phospholambins. They're right now they're not attached to it. They're just close to it, okay? The circuit is working 100%. When the first phospholambin attaches this circuit to a molecule, the circuit is going to start pumping calcium slower, in a lower amounts, slower, because one phospholambin molecule, which inhibits the circuit or like decreases its action, when one binds to it, the circuit is going to basically pumps calcium in a lower rate, in a slower rate, basically, okay? This is when one binds. When the second phospholambin molecule binds to the circuit, the pumping of the calcium is going to be even slower. So the circuit is weaker, basically. It's not pumping calcium as rapidly. Okay. When the third phospholambin binds to it, the circuit is even slower. It's like more and more slower. When the fifth phospholambin binds to the circuit is very slow to the degree it's almost not working. Is this clear? So when we want the circuit to pump the calcium back to the SR, the phospholambins detach from the circuit, so the circuit can go back to working. To work again. Is this clear? Is it really clear now, or do you want me to repeat again? The whole interaction between the phospholambin and the circuit. 100%. Okay, okay, you guys are saying that's clear. Like, so this phospholambin, okay, let me just go back to my notes. This phospholambin, what do you need to know about this phospholambin? This phospholambin is always attached to the circuit. In like normal case, we're talking about a normal muscle. This phospholambin is attached to the circuit, so the circuit does not pump calcium inside the SR. When does this phospholambin dissociate from the circuit? It's phospholambin, so it contains phosphate. So what do we use? We use something that we call phosphate kinase A. You probably heard about this kinase before. Have you guys heard about this before? Phosphokinase A, basically PKA. Have you guys heard about this before? Yeah, basically when we have a lot of calcium inside the cell, okay, this PKA becomes active, okay? This PKA, phosphokinase A, goes to the phospholambin and phosphorylates it, okay? So phospholambin, normally non-phosphorylated, it's attached to the circuit. When we phosphorylate it by PKA, it detaches. So the phosphorylated version of phospholambin is detached from the circuit, gets detached from the circuit. Is this clear? So PKA phosphorylates the 5 phospholambins, and the circuit now is free and it's pumping. Okay? When we don't want the circuit to pump again, to pump more calcium in the circuit, or like basically we don't have more calcium in the cytoplasm, so the circuit needs to stop working for the next contraction to happen. These phospholambins get dephosphorylated and they attach back to the circuit and the circuit stop working. Is this clear? Is the function of PKA clear here? And why do we need it? Yes? Yes, okay. So almost this is literally everything about in this slide. I know that I have like spent some time on it, but literally this is like the most important slide you need to know. Again, guys, heat review, action potential comes, basically moves here, DHPR. You need to know these two names, guys. Ryanodine receptor and the DHP receptor. You need to know these. Wallahi, you're gonna get asked about them. I can assure you, you're gonna get asked about them. 100%. No way you won't get asked about them. Like there are ABCs of this, so you need to know them. Basically, it gets inside the cell. Calcium gets inside the cell. It binds to the SR. Calcium is released. We have 100%, 30% from outside, 70% from the SR. The contraction happens. Now we need to get the calcium out. 28% we get it out by the exchanger, 2% by the calcium ATPase, and the circa is going to take the 70% that was released from the SR back into the SR. Should I move next, guys? Okay. Yes. Okay. So then let's move. Okay, now these are some like these next two slides are just like pure memorization. Guys, if just one thing I need to like I have to say now, if you guys just خلاص understood the last slide that we took, you خلاص almost understood like 90% of the lecture. Everything now is just like basically small things that you need like, to memorize. Basically, the similarities between the cardiac muscle and the skeletal muscle, both are striated, so you probably know that already. Striated, I mean, why they are striated, because we have integrating thin and thick filaments characteristics, A and I band. We talked about that. The thin filament circulatory proteins such as are present in both muscles. Remember, again, these are just similarities. You guys probably already all know all of this. And the cross bridge cycle is identical to the skeletal muscle. Why are we saying this? Because basically in the smooth muscle, it's different. It's not the same cross integrating. Integrating means like it's like they're above, like they're literally getting within each other. Integrating is like this. So the thin filaments, thick filaments, they're getting together. That is what is the meaning of integrating. Is it clear? Wait, I never went. Clear now? Okay. So now we need to know, okay, how do I get rid of this? Just let me hide the bar. Okay, never mind. Okay, now we need to know the differences. Again, guys, this slide, صراحة, there's not a lot for me to explain. It's just pure memorization. The T tubule is just wider than in the cardiac muscle. The T tubule, we said this. Remember how many times I emphasized on this point, because the doctor really like, said it multiple times. T tubule on the Z line. You need to know this. The mechanism of the SR calcium release is different. Why is it different? Because there's no physical interaction. Remember, skeletal muscle, there's literally physical interaction between the HPR and the ryanodine. Here, we don't have physical interaction. That's why it's different. Fewer DHPR in the card. Yeah, we said this, that there's lower amount. We talked about this too. Okay, this is just like صراحة, just know these are like two types. One type is in the skeletal muscle and one type is inside the cardiac muscle. The same, it's basically just like the same family, just different types. Like remember, Rhiannon 1 is skeletal, Rhiannon 2 is in the cardiac. Pure memorization. صراحة, not a lot to explain here. I'm gonna move next. Why wider reduces ionic depletion? It's not like it reduces ionic depletion. Oh, I'm gonna explain this. What? I don't know how I'm gonna explain this one. Uh, you see my number and my email at the start of the lecture. Please contact me there, and inshallah, I'm gonna reply to this question tomorrow, because like I have the concept in my head, but صراحة, like I'm not 100% sure. So I don't want to give you the wrong answer and then you go put it in the exam. So please just contact me, remind me, and inshallah I will give you the answer tomorrow, like whenever you send me the question. But please remind me. Okay, let's move next. So yeah, the question now. Calcium acts on what channel or receptor for the calcium 2 to flood out of the SR, to flood out of the SR into the cytoplasm? What is the answer, guys? Yeah, true. It is A. We said the ryanodine receptor. It is A. Do you want me to go over each single one of them again, or خلاص, you guys understood the concept? صراحة it's clear. Yeah, I think it's also getting late. Let's try to finish as صراحة as fast as possible. Okay, so not a lot is left, so don't be worried. Like a couple of slides. Okay, the sites of ATP usage. Guys, one thing you need to like again, this is something that you just need to memorize. Not a lot for me to explain here. Just my advice, or not my advice. It's like basically basic knowledge. Anything that has the word ATPs in it, that means it uses ATP. 100%. That means that basically it hydrolyzes ATP. Always. You see ATPs, it hydrolyzes ATP. So the circa, which I told you before, it's an ATPase. The calcium ATPase that we talked about, that basically pumps 2% of the calcium outside the cell. Okay, yeah. Yeah, I forgot to talk about Saraha. So remember, guys, when we said that we have sodium calcium exchanger that gets three sodium inside the cell and one calcium outside the cell, that basically contributes for kicking out 28% of the calcium outside the cell. Now we have three extra Na inside the cell, or three extra sodiums inside the cell. So right, we cannot have that because we just got them from outside. We cannot just keep them inside the cell. They're not normal, right? We just use them to get calcium out. So how do we get rid of them? We use the one that you all know, sodium potassium pump. You guys probably studied that in GI 100%. Like it's the most famous pump in the whole body. You know that 3 sodium out, 2 potassium in, and it uses ATP, right? You guys know the sodium potassium pump, right? I don't need to explain that, or do I? You guys probably took it in GI. You took it like in every single block by now. So yeah, basically the 3 sodium that we got from the exchanger, now we kick them out again by this pump because it's not normal. Them like exist like them inside the cell is not normal. That's why we need to get them out. This is an adenine cyclic molecule. It's almost like present in every single cell in the body. صراحة. Like it's always there. It basically converts ATP to cyclic AMP for the enzymes inside the cell and for like other like reactions to happen inside the cell. So obviously, since it turns ATP, it uses ATP. It converts ATP, so it's gonna use ATP, right? And obviously the main like the one that uses the most ATP and the one that uses the most energy is the myofilaments, the reaction. It uses 70% of the ATP that we need inside the cell. Okay, so I think this is very clear. Just you need to memorize the AT bases. Basically, if you memorized and like understood the pathway, you know this صراحة by default. The AT bases that we mentioned throughout the HATP. What HATP? What do you mean by HATP? You mean the sodium potassium pump? Or the calcium ATPs? The one after the adenine cyclase? The adenine cyclase. Okay, basically. Okay, guys, like this is something that you need to know صراحة in medicine in general. We have a molecule inside the cell that's called adenine cyclase. It's referred to by IC as AC, تمام? You guys probably took it. You took right now probably like since you reached second semester, you took multiple pathways right in the cell, and a lot of pathways. The starting point of it is cyclic AMP. So like you probably took that. So cyclic AMP is it's like it's a molecule, a secondary signaling molecule inside the cell. Just to get it, we use ATP. This adenine cyclase takes one molecule of ATP and converts it to cyclic AMP. Okay, so we can use the cyclic AMP in a lot of pathways inside the cell. It's just like it's not something that like really correlates to this pathway specifically that we just on like this calcium releasing pathway and how we get rid of calcium. It's just like something that's almost present in every single cell. That's why you need to know it. That's why you need to know that it uses ATP to convert it to cyclic AMP. You get it now. You know what cyclic AMP is, right? You guys studied that. Yeah, you studied that. Basically, just remember cyclic AMP is like a molecule that you're gonna always hear about. It's literally almost in every single path. Like it's in so many pathways inside the cell. So basically, we need to make it. We make it from ATP using adenine cyclase. Since we make it from ATP, it uses ATP because it's an ATP molecule that got converted. You see, yeah. Is it clear now? Okay. So let's move next. Again, this is just pure memorization. Man, I don't want صراحة, I don't know why the doctor put this in the lecture. This is small, but if the doctor wants you to know it, then he knows better. Just know these. In the adult heart, while he's fasting, what kind of energy we use? We use fatty acids. Okay. In the fetal heart, we use this like fetal heart. You know what fetal is, right? Basically, a small kid. Like not a small kid. Wait, I mean inside the womb, inside the belly of his mother, inside the belly of his mother. Like I cannot talk. Basically, the baby inside the while he's still not born yet, is the fetal. Their heart uses mostly glucose. Okay, and during fasting ketosis, basically ketosis, so you're gonna use ketone bodies. It's literally ketosis, so you're gonna use ketone bodies. You're fasting, you're producing ketones, so you're gonna use them for energy. During starvation, you use amino acids. Why is that? Basically, during starvation, guys, like your body just get into panic mode, and or not panic mode, but literally your body needs to get to make ATP. You know, to make ATP, you need to make ATP for the whole body, like to keep for you to stay alive. So the body start breaking everything off. So one of the things that your body break, other than fat and glucagon and glycogen, other than fat and glycogen, is proteins. And when your proteins are broken, they give you amino acids. Did you take it in mol 1, or is it something that you're going to take in mol 2? صراحة, I really cannot remember in which course it was. Did you take it in mol 1, or is it in mol 2? I think it's in mol 2. Basically, when you are like starving, your body breaks the protein to amino acids, which because proteins are made of amino acids, if you don't know that. And you start using these amino acids in the TCA cycle, which you probably already studied, and mol 1. Yes, you took it briefly. Yeah, exactly. So is this slide clear? صراحة, is you just need to know them. Yeah, like you just need to know like the adult heart while fasting. What does it do? You just know in each state what does the heart use? Because the doctor literally can ask you. He can tell you, someone like we got a patient to the ER. They are starving. What specifically the source of power the heart is using? Is it amino acids? Is it ketones? Is it you just answer. If he says starvation, then you put amino acids. If he tell you that the patient is in fasting ketosis, what does the heart use? It uses ketone bodies. It's just you need to memorize. Nothing صراحة a lot to explain here, so I'm just gonna move next. Okay, last part of the lecture. Guys, you remember in MSK we talk the length tension relationship? You remember that in MSK and the whole basically the whole relationship. You remember that, guys, when you took it in MSK? How in there, basically the more length inside the muscle, the more like tension it gets. Do you guys remember that? Uh, yes, but not much. Okay, so we're gonna just re-explain it. So this is, guys, the cardiac muscle, and this is the skeletal muscle. So these are different diagrams. Okay, let me just get the pointer so it will be more clear. This is the pointer. Yeah. So this is cardiac muscle, and this is the skeletal muscle. Where is the difference? Specifically here. You see how here it dips, here it doesn't dip. Why is that? We're going to talk about it now. So first of all, this is tension, this is length. The length is basically how much you stretch the muscle. The tension is with, just remember, the more that you stretch the muscle, the more length you give to the muscle, the more tension that's going to be made. in general. That is like in general, whether it's active tension, whether it's passive tension. Okay. Okay. So for active tension, what is active tension? Active tension is basically the tension that is made from a contraction, from the physical. That's why it's called active. Like the physical, not the physical. I mean like the contraction, the move that you put ATP into it to move it. Okay, that is the active tension at specific length, which is the perfect length for it, or like the normal working range as they call it here. Active tension is at its highest. Okay, is this clear? After this length, after this perfect length for the active for the contraction, like the perfect length for the contraction to make power, or like to produce basically power. After this, we're gonna start making something that we call a passive tension. This is obviously in the skeletal muscle. So this is the normal length of the muscle. Okay, at this length, the skeletal muscle will produce the highest active tension. So basically, the contraction is going to give you the most amount of power produced. So the contraction is going to be the strongest, basically. When you keep stretching the muscle, okay, after this point, the contraction won't be as strong. Okay, the contraction won't produce as much power. Is this clear? This is something from MSK, but at the same time, even though this active contraction is not stronger, you start basically making something that is called passive tension. Why is it called passive tension? Because it's like just you're not using any ATP into it. It's just the muscle is stretching and there is tension that is created from this stretch, from this lengthening of the muscle. So you see, after this normal range, the passive tension start getting up, okay? And the active tension goes down. Is this clear? This goes up, this goes down, and this is the purple one is the total. So the purple one is the sum of the both of the tensions. So basically, we add the passive tension and we add it with the active tension. We have this total tension thing. Okay, this is in the skeletal muscle. This is again in the cardiac muscle. This is the perfect working range. After it, we start having passive tension created, and active tension is going to go down. Okay, sorry. Okay, you don't need to be sorry, but yeah. Listen, you just need to know this. Let's think this is a muscle, okay? This is a muscle. To my two fingers. Are my two fingers here? Let me see the picture. Let me see camera. Yeah, this is a muscle, okay? Here right now, you see my tips of the finger. This is the perfect range for working, okay? Here, the contraction is not as strong when the muscle is shorter. Yani, look, you see here, the muscle length is shorter than the active, it's shorter than the perfect working range. So the muscle does not produce as much contraction. Like the contraction is not as strong. Okay. When you stretch and in the perfect point, it produces the strongest contraction, which is the highest active tension. Okay. After this point, the muscle is too stretched. Okay, the muscle is like getting more stretched than normal. So the contraction, when the contraction happens because of the stretching of the muscle, the contraction is just going to be weaker. That's why the active tension goes down, because you're not producing as much tension from the contraction itself. You get it now? So you need to be at the perfect working range. When the muscle is more stretched, the contraction won't be as strong because the muscle is not in the perfect position to be to get contracted. So since it's not in the perfect position, since it's not in the perfect position, the contraction won't be as strong. So the active tension produced from that contraction won't be as high. That's why it goes down. Is it clear? Okay. It's clear. Okay, nice. So remember, after this perfect range, we still have the passive tension going down because the muscle is okay. Let me get the paper again. So imagine this is the muscle, okay? This is the perfect working range. This is this is like we passed the length of the working range. We're still stretching it, so we're still trying to make it longer. So the passive tension is going to increase. You see, that's why it like skyrockets. Look how fast it goes up. The passive tension, it's just tension produced that we're stretching the muscle, but it's like we're stretching the muscle, but the muscle cannot get longer than this. That's why tension is getting higher and higher and higher. Okay, so that's the whole idea of the passive tension. What the doctor needs you to know here that why is there a dip in the skeletal muscle, but there's no dip inside the cardiac muscle? Okay, so I want you guys to think about the heart. Okay, when does the heart normally contract? The ventricle specifically. When the ventricle is full, right? When the ventricle is full, the ventricle contracts. So when the ventricle is full, basically the ventricle muscles are completely stretched because the ventricle is full. So the muscles inside the ventricle are 100% stretched. Okay. So when they are fully stretched, there is a signal from again from the SA node, the blood pumps, the heart pumps the blood. Okay. So right here, as you see, there is no going down for two reasons. One, because the normal range for the ventricle muscle or like for the muscles in the cardiac to work is fully stretched. So there's no more stretch to go. Here, the cardiac muscle is like this, not fully stretched. So when we increase the stretch, when we increase the length, we're outside the perfect working range for the contraction. But the muscle is not fully stretched, so the tension that is produced from the passive one is not enough. See, it's like not enough. So when active goes down, it literally goes down a little bit and then goes up. This is the first reason. The skeletal muscle, it's not fully stretched when it's contracting. There's still a little bit like there's the muscle is not fully like the perfect range is not when the muscle is fully stretched. Okay. On the heart, it's a different story because it's full with the blood. So when the ventricle is full of blood and the muscle is literally stretched, like the most it could be normally, like under normal physical conditions, this is the perfect working range when the ventricle muscle is like stretched the most. That is the whole idea. That's the first reason. So here, after the normal working range, we still have some stretch. The muscle is not fully stretched. So the total tension goes a little bit down. Here, no, it's fully stretched. So that's why the total tension would never go down because we have the passive tension compensating. Look, here it's higher than here. So it's basically compensating. This is the first reason. This reason the doctor really did not mention. I don't know why he really did not mention it. So I hope he does not ask you about it because it's a complicated concept. The second reason that the doctor mentioned, which is titin. Remember titin, the molecule that we talked about early in this lecture? You remember titin, don't you guys? Please tell me yes. Please say yes, you remember titin. The molecule that has like a spring that we talked. Yeah, the molecule that has like a spring. Basically, remember when we said it's like for elasticity, for when the muscle lengthens, then it goes back. Basically, we have different type of titans inside the skeletal muscle and different type inside the cardiac muscle. That's why we have here a dip. Here we don't have a dip. Is this clear, guys? I know this one's very confusing, صراحة. Like it really took me a lot of time for me to understand it too. So I wouldn't be surprised if you guys couldn't understand it from the first time. My advice, صراحة, if you did not get it from my explanation, I can re-explain it. Don't worry. Like if you want me to explain it, I would do it. But if you didn't understand it from me nor from the doctor, go watch some like the YouTube videos on it because like each person explain it in a different way because it's like kind of complicated. So it's like kind of weird. It's a weird concept. So there's a lot of videos on YouTube you can refer to if you couldn't understand it from my explanation or from the doctor's explanation. If you want me to repeat it now, I would 100% do it. So is it clear, guys, or what? Is it still a problem? Yeah, that's one of the two reasons. That's the reason that the doctor said. So that I think that is the reason that you need to specifically know. I mean, there's more salah than factors contributing to it, but that's the reason that your doctor has emphasized on in the recording. So I think that is the only reason you need to. I mean, salah, I don't want to give you my word and tell you the only reason that you need to know, but this is, I think, the most important one you need to know because your doctor emphasized on it. Okay, clear? Okay, I see clear in the chat. Okay, so I'm gonna move next. Okay, again, as we said in the previous one, as the EDV, you know what EDV is, right? And diastolic volume. So basically, when the heart is fully diastolic, is full with blood. As this increases, the stretch in the muscle is going to increase. So it's going to reach the maximum stretch. So basically, the contraction is going to be stronger. See, this is the stretch, this is the force. The more that you stretch the muscle, the higher the force gets, because you need a stronger contraction to pump the extra blood that is in the ventricle. Is this clear? So basically, in a cardiac muscle, the preload is the amount of blood inside the ventricle is the amount of blood. I mean, the EDV basically is the preload. Is this clear? I know, guys, this concept is kind of weird. صراحة, I know. Please repeat. Okay. Remember, guys, what we said before when I told you about the ventricle muscle, that it needs to be fully stretched to give you the strongest contraction it could be. Like the strongest contraction the heart is going to give you when it's fully stretched. Okay. I mean, when I'm saying fully stretched, obviously I'm talking about physiological levels. I'm not talking about pathological levels. So we're not talking about any specific diseases. We're talking about normal levels. Obviously, if you keep stretching the heart, obviously, then it's going to get out of the normal range and you're going to have a problem. We're talking about physiological levels. So what happens is here, as you know what end diastolic volume is before everything, you know what is that, right? If you don't, I'm going to explain what is it. No, okay, you don't know. That's the problem with this assignment, that with this lecture, because I think before introducing this concept, they had to introduce you, like, to explain what is the cardiac cycle is. They probably still didn't. But anyway, we're gonna explain it. So the end diastolic volume is, just notice now, that is, the heart is gonna pump blood, right? And then the heart is gonna refill blood for it to pump again. The ventricle is gonna get refilled with blood for it to pump. Okay, is this clear? You know this, right? That the heart pumps and then get refilled with blood. You know this. Okay, so the end diastolic volume is basically the amount of volume that we have inside the ventricle before it pumps. Literally before it pumps. This is the end diastolic volume. Basically the maximum volume we have inside the ventricle before it pumps. The last volume. Okay. So the heart, خلاص, we don't have any more filling. This is the end diastolic volume. So basically what it's saying here is, the more this volume increases, so the more blood inside the heart is, the stronger the contraction by the ventricle muscle is going to be to pump the blood. Because there's more blood, so obviously you need stronger contraction from the ventricle muscles to pump the blood. Is it clear now? So the more this blood inside the ventricle is, which is the end diastolic volume, the stronger the contraction will be, so the heart will be able to pump the blood. Because if there's more blood, because logically, if there's more blood inside the ventricle and you give it the normal contraction, not a stronger one, you give it like you give a contraction at like 70. You don't give a contraction at 100, okay? You give it at 70. Not all the blood is going to be pumped out, right? Because this force pumps out a specific amount of blood. So when you increase this amount of blood, you need a higher force, a higher contraction, a stronger contraction. So what they're saying here is, when you increase this volume, basically the muscle stroke is going to become stronger. The pumping is going to become stronger. The contraction of the ventricle is going to become stronger. Is it clear now? Is it actually clear or please, if it's not clear, I will just explain. Re-explain it again. Is it 100% clear? So just know, the more blood you have inside the ventricle before it pumps, which is the end diastolic volume, the stronger the pumping is, the stronger the contraction of the heart is going to be. That is the whole concept here. Just what is the stroke volume? The stroke volume is basically the amount of blood that is pumped by the heart. You're gonna know that in the cardiac cycle. That is the problem. I'm telling you guys, these slides right now won't make a lot of sense to you. But when you study the cardiac cycle and come back to this, you're gonna be like, oh, okay. Stroke volume is the amount of what is stroke? Stroke is each pump of the heart. So when we say stroke volume, it's the amount of blood that one pump of the heart is pumping into the aorta. Remember, the left ventricle pumps blood inside the aorta, right? Right? So the stroke volume is the amount of that blood that get pumped. So basically, they're telling you if there's more blood inside the ventricle, the force of the contraction is going to be stronger, which means that the amount of blood that's going to get pumped is higher than normal. Like the higher than like less amount of end diastolic volume. Okay. Okay. Well, okay. Let me just get a number. Maybe a number is going to become simpler. Okay. Let's say these are hypothetical numbers. Okay. Let's say you have 100 ml inside the ventricle. Okay. This is the first contraction. The second contraction, you have 150 ml. Okay. For this 100 ml in the first contraction, you need, for example, let's say, for example, you need 60, you need the force, you need 60 force, okay? The contraction needs to be at the strength of, as the strength of 60, okay? So basically, the heart is going to pump by the strength of 60, and the amount of volume that is going to be pumped inside its blood is the specific amount. Let's say the blood that's going to go, the stroke volume is going to be 70. Okay, let's say that 70 ml from the 100 ml are going to get pumped up into the aorta. Okay, so this is the stroke volume. This is the end diastolic volume. The force, basically, you know, how can we detect the force? We cannot detect how strong the contraction is, right? Like logically, there's no way that we can detect that. So we use the stroke volume, the amount of blood that was pumped into the aorta, as like basically as the measurement for how strong the contraction is. So the stroke volume is the amount. Okay, I'm probably complicated more now. I made it more complicated. Okay, forget everything. Forget everything that I said. The stroke volume is the amount of blood that get pumped into the aorta. Okay, from the ventricle. The ADV is the amount of blood that we have inside the ventricle. Okay, okay. Basically, what they're telling you here is the more ADV we have, the stronger the contraction of the ventricle muscle is, which means the more stroke volume, the higher the stroke volume is going to be. So the more blood pumped into the aorta is going to be. Okay? Because we cannot physically detect how strong the muscle is contracting, how do we measure it? We basically use the stroke volume, the amount of blood inside the aorta. So if the heart pumped 80 ml inside the aorta, which is the stroke volume, obviously it's going to be stronger than the heart who pumped. Obviously, I mean, the contraction is going to be stronger than a contraction that pumped only 50 ml, right? Logic. If you have 100, if you have 100 in each ventricle, one pumped 80, one pumped 50, which one is the strongest? Which one is stronger, I mean? The one that pumped 80, right? Is this clear? I think I'm pretty sure you lost. I'm pretty sure you lost, Saraha, and I don't blame you. Yes, it's clear? Oh, no, I got it. Okay. Even if you didn't get it 100%, you're gonna get it inshallah more when you study the cardiac cycle. Saraha, teaching you this before teaching you the cardiac cycle is not something that should have been done, but I guess you gotta live with it. When you're welcome, Uh, when you get when you basically finish the cardiac cycle, which is almost the most important like lecture in the whole cardiophysiology, this is gonna make way more sense. Because like there's so many things you don't know still about how the heart pumps blood and whatever. That's why it's still like weird me telling you this pumps this and this amount and this amount. Okay, let's just get with the definitions at least here, and inshallah after you get the cardiac cycle, you're gonna understand this more. So this relationship, as we said, which is the increase in ADV means stronger contraction, is known as the Frank-Starling law. Just know the name of the law, Frank-Starling law, which is the degree of the stretch on the heart increases the force. Remember, we said that when we have higher volume inside the ventricle, that means that the ventricle is going to stretch more. So we need more force to pump that blood. That's why the contraction force is going to be higher. Clear? Should I move next? Tamam. Let's move next. Okay, what is the afterload? Again, would have made way more sense if it's after the cardiac cycle, but You took it before the cardiac cycle, so we gotta explain it. Guys, the afterload, as you probably took in MSK, it is what you're working against. So it's basically what you're trying to achieve. So if we're talking about someone who's going to the gym, okay, you're in the gym and you want to lift some weight. For your biceps to lift the weight, this weight is the afterload. So your biceps need to produce a contraction and a force that is strong enough to lift this weight. Okay. So what is the afterload? It is the weight itself. It is what you're working to achieve. It is what you're working against. You're pushing against the weight up, right? So you can lift it. So this is the afterload. Is it clear in the case of the biceps and the skeletal muscle? Okay, it is clear. For the heart, the afterload, or what you're working against is the aortic diastolic pressure. What is the aortic diastolic pressure? Basically, normal case, the ventricle is filled with blood, right? Why does not that blood just go directly into the aorta with no pumping? Because the pressure inside the aorta is higher than the pressure inside the ventricle. If the heart is not pumping, that's why the blood does not go up. Why does the heart pump? Why does the heart produce this forceful contract, like this strong contraction? To make the pressure inside the ventricle higher than the pressure inside the aorta. So when the pressure inside the ventricle is higher than the pressure inside the aorta, the blood moves from the ventricle to the aorta. So what is the afterload? Basically the pressure inside the aorta. That is what you're trying to go higher than. The whole contraction point is just to pass this number, to make the pressure inside the ventricle higher than the pressure inside the aorta. This is the whole concept of the afterload regarding the heart. Is it clear? Just if you get a question, what is the afterload regarding the cardiac muscle? Just write aortic diastolic pressure. Again, would make way more sense after you study the cardiac cycle, believe me. If it does not make more sense after you study the cardiac cycle, come find me. I can assure you it will make way more sense. Okay, last slide. For this slide, again, it's a weird concept. It's actually a concept that I find very hard also to understand. So the doctor said a couple of things, and I advise you just to know it. This is what we have, what we call the shortening velocity, and this is the increasing preload, and this is the afterload. Okay. I mean, no. What did I say? Okay, let's look at this diagram first. Forget everything I said. My brain is Salah turning off right now. So this is shortening velocity. Okay, the shortening velocity. Shortening velocity means how fast the muscle becomes short, like how fast the contraction is. Okay, and the afterload, which is the force that we're working against. Again, we said it. The force that we're working against is the afterload. So basically, when the shortening velocity is at maximum, so the muscle is contracting super easily, that means that the afterload is that it's zero. There's no afterload. Like if I do this, there's no afterload, my muscle will contract easily. But if I try to lift, for example, this cup, there is an afterload. So my muscle won't shorten as fast, won't contract as fast. Is this clear? Is this clear? Okay. Type. The one thing that the doctor wants you to know here, that when you increase the preload, okay, so you're increasing the preload in this case. When you increase it, the shortening velocity also increases. So basically when the heart, we said the preload in the case of the heart is the amount of blood inside the ventricle, right? So when you increase the amount of blood inside the ventricle, the shortening velocity is going to increase. So the heart is like the ventricle muscles inside the heart is going to take more time for them to contract. Very small differences, obviously, but it's just a concept that you need to know. Preload and afterload. Okay. Let's go back again. Okay. Preload. Remember, when we said in the case of the heart, the preload is the amount of blood that is inside the ventricle. Remember when we said this? EDV, this is the preload. In the case of the heart, the preload is what we work with, is like basically what we have, the amount of blood. The afterload is the force that we're working against. So the force that we're trying to become higher than the force that we're trying to achieve, which is the aortic pressure, the pressure in the aorta. So the preload is the amount of blood inside the ventricle. In the case of the heart, the afterload is the pressure inside the aorta. Is it clear? Okay, nice. So basically what they're saying here is, okay, what they're saying here is when you increase the preload, so when you're increasing the amount of blood inside the ventricle, the shortening velocity, yeah, which is how fast the muscle is gonna contract, or how fast the muscle is gonna become shorter, takes more time because you have more blood inside the ventricle. So if you have more blood inside the ventricle, obviously the heart would take more time for it to pump, for it to contract. Obviously, it's not as fast. I mean, obviously the differences are very small, but you still need to know this relationship because you can get asked, in the exam. How will the question be? Probably the question is going to be like this. They're going to give you this diagram and tell you that the purple one is the normal one. Okay, is the normal is the normal preload. I mean, the purple one is the shortening velocity at the normal preload, and then we increase the preload. Which one is the new shortening velocity curve? One or two. When we increase the preload, it basically just moves to the right, so it's going to be one. You get the concept? This is probably 90%. This is how the question is going to be. I don't think they're going to go more complicated than that because صراحة, I'm second year and I never studied anything more complicated than that. Okay, is this good? Okay, yes. So yeah, with this I conclude, I guess. This is the last slide. We have a couple questions we can solve. I obviously want to thank the two people who صراحة were fighting till the end. I hope that I made the lecture easier on you. صراحة the physiology first couple of lectures are kind of mess. You just new to the block and a lot of things are going on. So I hope I did a good job explaining it. Do you have any questions about my whole part now before we get into the questions? Any questions, guys? I mean, there are only two. Well, do you guys have any questions? Any point? Okay, you don't. I mean, the other guy is not replying, or the other girl is not replying. So I guess. Okay. Oh, they reply. Okay, so they don't have a question. Nice. I hope so. Let's start solving the question. There are like two questions, I think, or three. So they're very simple. I'm gonna let you guys read the question and you answer me. Let's see if you got the concept right or not. Is it D? Let's see. It is D. Good job. Did you get why is it D, or you just threw it? It's just the law. Basically, you memorize the law. Good job. I get it. Nice. 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 Okay. The next one. Okay, this one is a long scenario. صراحة, it's the one that I created myself. I did not get it from a book. I just want you guys to get used to what a scenario is. So yeah, take your time. You don't need to answer it in 2 seconds. Good job, guys. You may be proud, صراحة. After these 2 hours, you guys actually understood. Very good. Good job, people. Wallahi, very proud of you. Nice. It is 70% indeed. That is true. And yeah, thank you guys for attending. Really, thank you. I really appreciate you guys staying here. I really appreciate the coordinator and the the girl who helped us. Thank you so much for staying this late. Sorry, I know we took way more than we should be.